Everything around us is made of cells, from the tiniest bacteria to the largest trees. But what exactly is a cell? How do scientists study them? And why is the cell called the fundamental unit of life? Today, we'll break down the entire Insert Class 11 Biology chapter Cell, the unit of life. Discovery of the cell. Robert Hooke, 1665, observed cork under a primitive microscope and coined the term cell. Anton van Leeuwenhoek, first to observe living cells, bacteria, protozoa. Schleiden and Schwann, proposed cell theory. 1. All living organisms are made of cells. 2. Cell is the structural and functional unit of life. Rudolf Virchow, 1855, added, Omnis cellula e cellula, all cells arise from pre-existing cells. Types of cells. Prokaryotic cells, simple, no true nucleus, no membrane-bound organelles, e.g., bacteria, archaea. Eukaryotic cells, complex, with true nucleus and membrane-bound organelles, plants, animals, fungi, protists. Differences: size, complexity, DNA form, circular versus linear, ribosome size, 70S versus 80S. Size and shape of cells. Cells vary greatly in size. Mycoplasma, 0.3 m, smallest cell. PPLO, 0.1 m. Bacteria, 1 to 2 m. RBC, 7 m. Eukaryotic cells, 10 to 20 m. Ostrich egg, the largest cell. Shapes. RBCs, round, biconcave. WBCs, amoeboid. Nerve cells, elongated, branched. Columnar epithelial cells, long and narrow. Prokaryotic cells. Bacteria are prokaryotic cells, meaning they lack a true nucleus and membrane-bound organelles. Let's break down their key parts. Pili, long, few in number, about 2 to 5, used mainly for conjugation, that's bacteria's way of sharing DNA. Fimbri, short, numerous, 200 to 300, help bacteria attach to surfaces. Ribosomes, 70S, protein-making machines inside the cell. Flagella, for movement. Depending on arrangement. Monotrichous, one flagellum. Amphitrichous is flagella at both ends. Lophotrichous, tuft at one end. Peritrichous, all around the cell. Polysomes, clusters of ribosomes working together. Inclusion bodies, store nutrients like glycogen, polyphosphate, and proteins such as cyanofissin granules. Mesosome, enfolding of the cell membrane, helps in respiration and cell division. Genophore, the bacterial chromosome. DNA. Glycocalyx, protective outer coating. Capsule, hard, sticky, protects from drying and immune attack. Slime layer, loose and slippery, helps in movement. Cell wall, made of peptidoglycan, gives shape and protection. Cell membrane, selectively permeable, controls what enters and exits. The flagellar structure includes filament, long tail-like part. Hook, connects filament to basal body. Basal body, anchors the flagellum in the cell wall and membrane. Gram staining. To identify bacteria, microbiologists use gram staining. 1. Crystal violet stain, both gram-positive and gram-negative bacteria turn violet. 2. Iodine, mordant, fixes the dye inside the cell. 3. Alcohol wash. Gram-positive, retain the violet dye because of their thick cell wall. Gram-negative, lose the violet dye due to their thin cell wall. 4. Safranine stain, counterstains gram-negative bacteria pink. Results. Gram-positive purple, gram-negative pink. Cell membrane overview. The cell membrane is not just a boundary, it's a selective gatekeeper. It's made up of a lipid bilayer with embedded proteins, glycoproteins, and glycolipids. These molecules maintain membrane stability. In animals, cholesterol does the job. In plants, it's phytosterols. In fungi, ergosterol. Proteins mainly move laterally thanks to the quasi-fluid nature of the lipid bilayer. Transport across membrane. Transport is of two main types. 1. Passive transport moves substances from high concentration to low. No ATP needed. Diffusion, direct movement of small molecules. Facilitated diffusion needs transport proteins like channels or carriers. 2. Active transport moves substances from low concentration to high. Requires ATP and transport proteins. Example, Na/K pump. Bulk transport. When the cell moves large molecules, it uses bulk transport. Exocytosis, inner to outside. Also called cell vomiting. Endocytosis, outside to inner. Includes phagocytosis, cell eating, forming a phagosome. Pinocytosis, cell drinking, forming a pinosome. 2. Cell wall, plants, cellulose, hemicellulose, pectin, middle lamella, calcium pectate. 3. Nucleus, double membrane, nuclear pores, nucleoplasm, chromatin, euchromatin, heterochromatin, nucleolus. Endomembrane system. The endomembrane system is a network of functionally connected organelles, all single membrane bound, like the Golgi, lysosomes, and vacuoles. Endoplasmic reticulum, divides the cytoplasm into luminal, inside, and extraluminal, outside, compartments. It has three forms. Cisternae flat and interconnected, represented by RER. Has ribosomes for protein synthesis. Tubules, cylindrical, represented by SER. No ribosomes, used for lipid synthesis. Vesicles, round, transport materials inside cells. Rough RER, has ribosomes and ribofurans, makes proteins, lysosomes, enzymes, and even forms SER. Smooth SER, no ribosomes, makes lipids, like sex hormones, forms Golgi bodies, detoxifies drugs, and helps CA superscript 2 movement during muscle contraction. Golgi apparatus is packaging, secretion, lysosome formation. Lysosomes, digestive enzymes, suicidal bags. Vacuoles, storage, turgor pressure maintenance. 5. Mitochondria, double membrane, inner folds, cristae, ATP synthesis, have own DNA and ribosomes. 6. Plastids, plants, chloroplast, photosynthesis, chromoplast, pigments, leucoplast, storage. 7. Ribosomes, protein synthesis, 80S in cytoplasm, 70S in organelles like mitochondria and chloroplast. 8. Cytoskeleton, microtubules, microfilaments for shape, movement. 9. Cilia and flagella, movement, 9+2 arrangement of microtubules. 10. Centrosome and centrioles, cell division in animal cells. From the microscopic bacteria to the complex human body, the cell is at the core of life itself. Keep exploring, because the more we understand cells, the more we understand life. Don't forget to like, share, and subscribe for more biology made simple.